the biotech was created in 2020 about a discovery we made a few years ago, which is that natural killer T cell can be activated by protein antigen. The point is that the lineage we have identified is activated by peptides, amino acid stretches, hydrophobic peptide as a matter of fact. So all of a sudden, you have a possibility to interfere with the immune system, in the good sense of the word, interfere with an antigen specific for, let's say, an infection, a tumor, and so on, and so on. So the second characteristic of this lineage is the fact that it is not purged out, is autoimmune reactivity in the thymus. So it's created by the bone marrow, transit to the thymus, where it has two phases of expansion, but there is no elimination of autoreactive T cells, meaning that in the periphery, this is true in human beings and in the mouse, you have a plentiful of possible specificity, which can be autoantigen or, of course, alloantigen by definition. So all of a sudden, you have a population of cells capable of reacting against whatever you can imagine in the universe. This is, of course, pretty important because then you have a series of cells on which you can do whatever you like. Activate them in oncology or infection, for instance, or eliminate this activation in other settings like replacement factor therapy, where the immune response against the factor used as a replacement factor is eliciting an immune response. So we have identified a number of possible epitopes, and indeed we have identified in several individuals and in the mouse, of course, the presence of NKT cells reacting against the mutated P53. So the tools are there. Now, the point is, well, let's say twofold. The first one is to test whether the cancer cell does express CD1D so that we have something to present to the NKT. Well, the news are quite good because if the T cell, if the tumor cell is not expressing CD1D, you can force it to express CD1D with epigenetic inhibitors or with even vitamin A or very simple, and irradiation, as a matter of fact. This is extremely potent. If you like, CD1D is expressed in stressed cells. And of course, by definition, tumor cells are stressed because they have to multiply very quickly and so on and so on. It's interesting to know that there are publications showing that NKT can also modify the TME, the tumor microenvironment, by activating essentially dendritic cells and macrophages and turning some of the macrophage from the phenotype, which is called type 2 to type 1, which is, capable of activating, again, CD8 cells and so on. Again, you create a kind of cascade event, which is a virtuous cascade, creating an environment, not only in the tumor cell, but also in the environment. The next step is, of course, to take a humanized mouse model, put a tumor cell from human beings, and see whether or not we can eliminate the tumor using NKT cells, either by vaccination or by transferring NKT cells from another source.